Good morning, everyone. My name is Abigail Zaroff, and I am a rising sophomore at Duke University and a summer trainee in the Mayan Lab at the Icahn School of Medicine at Mount Sinai. Today, I'll be presenting about the project I worked on this summer, which focuses on crossing omics datasets from human cell lines for knowledge discovery. Before diving into the project, it is important to discuss some concepts about cell signal pathways. Cell signaling pathways form communication networks within cells. So when a ligand binds to a receptor at the cell surface, it triggers a series of intracellular events known as signal transduction. This leads to the activation or inhibition of specific proteins and results in a cellular response. In cancer, as illustrated in the diagram, mutations in cell signaling proteins break cell signaling pathways, leading to uncontrolled cell division and proliferation. The datasets used in this project offer valuable insights into how the cell signaling pathways function inside human cell lines. The datasets used in the project were extracted from several recent seminal publications in the field of systems biology. The first paper explores basal protein expression levels across a diverse set of cancer cell lines. The second paper analyzes the proteome response to perturbations with 875 compounds in cancer cells. The third paper introduces the kinase library, a study that discovered the substrates of many human serine threonine protein kinases. The fourth paper represents the Lynx L1000 data. Lynx L1000 data contributed two datasets to the project. One is a transcriptomics response of cancer cell lines to chemical perturbations, and the other to single gene CRISPR knockout. And the last paper is from the DepMap project. The data from this paper is about the effect of gene knockouts on cell viability and proliferation of cancer cell lines. Each of these papers contributes towards our understanding of cell signaling pathways, adding another dimension to accumulating evidence about how these pathways are functioning as a network. This figure is taken from a review article published by the Mayan Lab titled Lean Big Data Integration in Systems Biology and System Pharmacology. It represents how systems biology data collected by different methods can be integrated. By converting the data into networks, gene set libraries, or bipartite graphs, we can combine the data for synergistic discoveries. Following the ideas from this review article, the first step of this project was to convert the data sets from the papers into gene set libraries. Luckily for me, almost all the data sets were already converted into gene set libraries except for the DepMap dataset. The fact that the DepMap dataset was not ready was also great for me because I was able to learn how to process it and create a gene set library from it. So before starting integrating the data, it is important to fully understand the contents of each individual gene set library. So let's review these again. The kinase library provides information about kinases and their substrates. Lynx L1000 chemical perturbations provides information on the effects of drugs and small molecules on gene expression. The Proteomics Drug Atlas created by the Gigi Lab from Harvard Medical School provides information about the effects of drugs on protein expression. Lynx L1000 CRISPR knockouts dataset provides insights into the effect of single gene knockouts on gene expression. CCLE Proteomics provides basal protein expression in cancer cell lines. And finally, DepMap provides information about how single gene knockouts affect the viability of cancer cell lines. The next step was to begin to cross pairs of libraries. Each gene set from a specific library was compared to another gene set from a second library, using the Fisher exact test and Jacquard method to assess overlap. The set pairs with the most overlap based on the Fisher exact test were output into a file and investigated further. This diagram shows how the various datasets are related. For example, by combining the Lynx L1000 drug perturbations and Proteomic Drug Atlas, we can look for concordance across these two datasets. That is just one example of the value of combining these datasets. This is an example of a table that lists the top 5 gene set pair overlap between all library pairs. As you can see, the columns include the library names, gene set names, the Jacquard index, the Fisher exact test p-value, the overlapping genes, and the number of overlapping genes. The same information from the table was used to create a network diagram showing the connections between gene sets that had the greatest overlap. The network is color-coded by library, and it provides a way to see the information from the table more clearly and identify clusters of sets with high overlap. Next, we asked how many of these connections were already known. To do this, we utilize PubMed. We search for each gene set term individually and then together. The bar graph shows the top 10 gene set pairs with the highest PubMed literature co-mentions. Next, we wanted to focus on some highly scored connections that did not have any literature support. This network diagram shows the overlapping genes between the Lynx L1000 CRISPR knockout gene SEMA4G and the HepG2 cell line from the CCLE proteomics dataset. There are 39 overlapping genes from the two gene sets, which is highly significant. The HepG2 is a liver cancer cell line, and the SEMA4G is a protein that is known to serve an axon guidance by acting as a receptor for plexin B2, which is also known to play a role in cancer progression. However, there are only a few publications about SEMA4G, so its function in the liver is mostly unknown. As you can see, it is highly expressed in the liver, and functional predictions based on gene-gene co-expression using the tool PRISM-Exp, recently published by the Mayan Lab, suggests that it plays a role in fatty acid metabolism. This is further supported by enrichment analysis applied to the 39 overlapping genes between SEMA4G knockout down genes and the HepG2 cell line basal proteins. GEO Molecular Functions 2023 and Bioplanet 2019 both show enrichment for terms related to fatty acid metabolism. In conclusion, combining various related datasets collected from human cell lines can aid in the discovery of new knowledge, and this can lead to the discovery of new drugs and targets for cancer and other diseases. For future directions, first, the libraries can be combined in sets of 3, 4, 5, and 6 for further analysis. In addition, the compiled data can be stored in a database to provide researchers easy access to the data, enabling efficient querying and further exploration of potential novel associations. And lastly, I would like to give a big thanks to the entire lab for all their unwavering support. I'd like to extend a special thank you to Dr. Mayan, Ms. Jenkins, Ito, Nash, and Sherry. Thank you for this opportunity, and thank you for listening. Please put any questions into the Q&A, and now the next speaker, Garvi Awasthi, will present a method that predicts immunotherapeutic targets for non-brain solid tumors.